The process of making blood cells or forming blood cells is called hematopoiesis. Before you're born, hematopoiesis occurs first in the yolk sac of that developing embryo, and then later in the liver, the spleen, the thymus, and the lymph nodes of the developing fetus. The red bone marrow becomes the primary site for hematopoiesis in the last 3 or so months before a baby is born, and that continues to be the place where hematopoiesis occurs throughout the rest of your lifespan. So red bone marrow occupies the microscopic spaces between the trabeculae and your spongy bone. It's present in the bones of the axial skeleton, the pectoral and the pelvic girdles, and the proximal epiphyses of your humerus and your femur. Do you remember what epiphyses are? That's right, that's the end. Which ones are proximal? The ones closest to the point of attachment. So in that red marrow, there are cells called pluripotent stem cells, or hemocytoblasts. You may also hear them referred to as hematopoietic stem cells. That all means they're stem cells that have the potential to develop all the different types of blood cells. So this is a pretty complicated little image here, but we're going to try to make it a little bit easier to understand. So remember, in your bone marrow, you have stem cells that reproduce themselves, proliferate, make a whole lot more, and then differentiate into cells that are going to give rise to all of the blood cells. So if you look at the very top of the image, you see that pluripotent stem cell. Remember, that's in the red marrow. So it can produce 2 different types of stem cells. Each one of those stem cells has the capacity to develop into several different types of cells. So here's our pluripotent stem cell. It can divide and become a myeloid stem cell or a lymphoid stem cell. So this pluripotent stem cell has the ability to differentiate or specialize into either one of those cell types. There are several different things that can happen there. So the myeloid stem cells give rise to the red blood cells and the platelets, the monocytes and neutrophils and eosinophils and basophils and mast cells. Yeah, a lot going on there, right? The lymphoid stem cells simply give rise to the 2 different types of lymphocytes, okay? Or the different types of lymphocytes. So here we see the pluripotent stem cell differentiates to become a myeloid cell, which can give rise to red blood cells, platelets, eosinophils, basophils, neutrophils, and monocytes. So basically everything except the lymphocytes, because your natural killer cells are a type of lymphocyte. So your B lymphocytes, your natural killers, and your T lymphocytes all result from that lymphoid type stem cell. Wow, there's a lot going on there, huh? So how do these stem cells know what to do? Well, there are several hormones that are called hematopoietic growth factors, and each one of those regulates the differentiation and proliferation of different types of cells. So there's a hormone called erythropoietin. We talked about erythropoiesis just a minute ago. Remember, erythropoiesis, or hematopoiesis, was making of blood cells. So all of this that you see here is hematopoiesis. But erythropoiesis is when these myeloid stem cells make erythrocytes. Leukopoiesis is when this stem cell is going to be stimulated to make leukocytes. Well, again, that's all well and good, but how does the stem cell know what to do? So if your blood oxygen level is low, your kidneys are going to secrete erythropoietin, a hormone. Remember, erythro means red. So erythropoiesis causes red blood cells to be made. There are several things that are going to stimulate white blood cells to be made. There are colony stimulating factors and interleukins, and all of those are cytokines, but they're different kinds of cytokines. So depending on what the body needs, if you need more eosinophils, because your eosinophils are going to destroy parasites, then a certain colony stimulating factor is going to be secreted, and you're going to make more eosinophils, and the same thing for all of the other types. So the specific chemicals, the hormones, the thrombopoietin if you need more platelets, the cytokines, which include your colony stimulating factors and interleukins, all of those tell that hematopoietic stem cell, make more of this type of cell, because here's what's wrong in my body and this type of cell can fix it. Make sense? All right. Now that we know a little bit about where those cells are made and the different things that trigger it, let's talk a little bit about the individual cells. So your erythrocytes are your red blood cells. Now, they're not true cells when they're mature and doing their job. They begin as true cells when they're created from those hematopoietic stem cells. But they have a very simple structure. They're biconcave, which means they're sort of squished like a donut that doesn't have a hole all the way through, and they're basically just little bags of hemoglobin. Now, hemoglobin is the molecule that's going to carry oxygen, and globin refers to protein. Hemo refers to the iron part of the hemoglobin. So if you look carefully at this hemoglobin molecule, you can see there's a light blue chain and a light blue chain and a dark chain and a dark chain. Well, what that really is 4 different protein chains, polypeptide chains. And do you remember how we make polypeptides, how we make proteins? We hook amino acids together to make a long chain structure, right? Remember, all of that is determined by your genes, and your genes determine how we put the amino acids together. So that's what happens with hemoglobin. We make 4 chains of proteins. We make 2 chains that are identical, and those are 2 beta chains, in this image, the dark blue ones. And we make 2 chains that are identical. In this image, they're the light blue chains. Those are the alpha chains. Now, each one of those is a polypeptide, but they all hook together to form functional hemoglobin. So hemoglobin is a quaternary protein. Remember, that's where you have more than one amino acid chain that works with another one. So each one of these amino acid chains, all 4, bind to one iron. And a part of that is the heme. So 4 polypeptide chains, each one binds one hemoglobin. So that means that each hemoglobin can bind 4 oxygens, because your oxygen molecules bind to that iron. So I mentioned that erythrocytes are not true cells. And the reason that they're not true cells is because when they're functioning, they don't have a nucleus. But where'd all that hemoglobin come from? What do you have to make a protein? You have to have a gene. What do you have to have genes? You got to have a nucleus containing all that DNA, right? So way back early in that development process that we looked at just a minute ago with hematopoiesis, these erythrocytes have a nucleus. And very early in their development, they start making lots and lots and lots and lots of hemoglobin. Once they become basically just a great big bag of hemoglobin, then the nucleus is ejected. Once that nucleus is ejected, that makes more room for carrying more hemoglobin and carrying more oxygen. Because these mature red blood cells don't have a nucleus, all of their internal space is available for transporting oxygen, but they also lack a lot of organelles, including mitochondria. Since they don't have mitochondria, they don't use up all that oxygen they transport. They can simply take it out to body cells so that body cells can then use that oxygen to make lots and lots of ATP. Each erythrocyte contains about 280 million hemoglobin molecules, and each one of those hemoglobin molecules can carry 4 oxygens. Yeah, you can do the math and figure out how much oxygen can be carried in one erythrocyte. So I want to do that math a little bit further. Let's think about that. A healthy adult male has about 5.4 million red blood cells per microliter of blood. Now, remember, 1 microliter, there's 1 million of those in a liter. So 5.4 million red blood cells in 1 microliter. And the healthy adult male has about 5 liters of blood. That's a lot of erythrocytes. That's a lot of hemoglobin. That's a lot of oxygen. Okay? So 1 drop of blood is about 50 microliters. Imagine how many oxygen molecules are in that 1 drop. So to maintain normal numbers of erythrocytes, we've got to constantly be making new cells, getting those new cells into circulation to balance out the way that red blood cells are destroyed in the body. Red blood cells have a very short lifespan because if you don't have a nucleus, you can't make repair proteins, right? A lot of the capillary beds throughout the body are very, very small, and only 1 erythrocyte at a time can pass through those capillary beds. So some of them sort of have to do like Fat Man Squeeze, if you've ever been to the rock formation up in Lookout Mountain in Tennessee. So it's basically 2 really, really tall rock walls that are very, very close together. And you have to squeeze as you make your way through that passageway. That's the way these erythrocytes have to do. So they're very, very small, but some of the capillary beds are even smaller. So they have to deform their cell membrane in order to get through those teeny tiny little capillaries. All of that puts a toll on that cell membrane. Just like our bodies wear out as we use them more and get older, erythrocyte membranes wear out. They can't repair themselves, so they have a very short lifespan. Most erythrocytes only can survive in the body for about 120 days, so they have to be we're constantly replenishing that supply. So what happens when the erythrocytes are old and broken down? Yeah, we break them apart and we recycle them. So old cells are removed from circulation by the spleen and the liver. So here we've got old erythrocytes. Remember, they're made of proteins, globins, and heme. Some of that heme is going to be broken down into biliverdin. The iron can be recycled and used again, taken back out and used to make new erythrocytes. Some of that, the biliverdin, will be converted to bilirubin. Then the liver continues to break it down into different chemical components, and you don't have to know all the steps, just get the idea of what's going on here. But the final component of the breakdown of this biliverdin and bilirubin becomes a pigmented chemical structure that gives some of that darkish, yucky looking brown color to the feces. Some of it will be released in the urine, and again, some of the iron and other proteins, some of the amino acids, will be recycled and used again in the body. Okay, as I'm getting ready to finish up on recycling red blood cells, I forgot to tell you one other thing, so let's back up here to hemoglobin for just a moment. Hemoglobin, as we talked about, transports oxygen out to body cells, right? We only breathe oxygen into the lungs. So the erythrocytes have to pick up that oxygen in the lungs and transport it out to body tissues. Once that oxygen, that blood, is out in the capillaries of body tissues, that oxygen dissociates or breaks away from that hemoglobin molecule. When it does so, then the tissues, of course, can use that to create ATP. But one of the byproducts of creation of ATP is carbon dioxide. We got to get rid of that, but how do we get rid of carbon dioxide? Of course, it's a simple question, right? We exhale, but we don't have lungs that can exhale in our intestines, or the cells in our little toe, or the cells in our ears, right? So all of those cells are making carbon dioxide when they're creating ATP, and that's a waste product that we've got to get rid of. So about 23% of that carbon dioxide, CO2, is going to be sort of brought in and attached to those hemoglobin molecules once the oxygen's gone. So some of the CO2 binds to the hemoglobin, but the rest of it is carried in the blood plasma as carbonic acid or as bicarb ions. So blood flowing through those tissue capillaries picks up the CO2, and as the blood flows back to the lungs, the CO2 is released from the hemoglobin, and then we exhale. All right, sorry about that, but I had to back up so I wouldn't forget to tell you that. All right, now we're going to talk a little bit more about the regulation of erythropoiesis. So remember before we talked about hematopoiesis, making all of those red blood cells. Well, let's focus specifically on making all of those blood cells, different types when we mentioned it a while ago. Let's talk specifically now about making red blood cells. So erythropoiesis is the action of, or the production of, making those red blood cells. And this in the adult, of course, occurs in the red marrow, as we mentioned earlier, specifically in the adult in the areas of the proximal epiphyses of some of your long bones. So we talked about red blood cells being recycled, but sometimes we just need to make new erythrocytes. So erythropoiesis, or making of red blood cells, and red blood cell destruction usually proceed about the same pace. So we break down about as many cells as we're making new cells to replace them. However, if your oxygen levels get low, the oxygen carrying capacity of your blood falls, maybe erythropoiesis is not keeping up with the rate that we're breaking down red blood cells. So negative feedback is going to kick in and cause your body to step up the way that it's producing red blood cells. So this oxygen deficiency out at the tissues is called hypoxia. So this may occur if there's not enough oxygen getting into the blood. For instance, if you go mountain climbing, at those high altitudes, there's less oxygen in the atmosphere. So less oxygen is binding to that hemoglobin, therefore less oxygen is getting out to body tissues, causing hypoxia. That oxygen delivery deficit may also cause a condition known as anemia. Anemia is not low iron. Let me repeat that. Anemia is not low iron. Remember, the iron in the hemoglobin binds to the oxygen. So low oxygen, that's anemia. Low iron can cause one kind of anemia, because if you don't have enough iron, you can't make functional hemoglobin. If you don't have functional hemoglobin, you can't carry enough oxygen. Okay? So the kidney's role in the body is to filter the blood. So as the kidneys are filtering the blood, if they detect hypoxia, the kidneys are going to be stimulated to secrete high levels of erythropoietin. Erythropoietin then travels through the bloodstream to target cells in the bone marrow. That's going to stimulate those stem cells in the bone marrow to make erythrocytes. Okay, so erythropoietin causes erythrocytes to be made. So as more and more erythrocytes are being made, they begin, remember, as those immature cells that have to make lots and lots and lots of hemoglobin. So those are reticulocytes. So those immature cells enter circulation and they mature in about 1 to 2 days. So that gives them a couple of days to stock up on making all that hemoglobin, eject those nuclei, and then be functional erythrocytes that are traveling throughout the body and carrying more and more oxygen. So as those tissues are getting more and more oxygen, then we return back to normal levels of tissue perfusion, tissue oxygenation. So hypoxia, low oxygen, is no longer a factor, so the kidneys now detect, okay, blood oxygen's back to normal, and they shut down that excess production of erythropoietin. Bam, homeostasis. Bam, negative feedback. Isn't the human body just amazing? All right, so that's what happens when things go right. But sometimes things don't go right. So we have homeostasis imbalances when we can't fix things as we normally would. So there's a condition called polycythemia. Okay, poly, many, cyte, cell, emia always means condition of the blood. So polycythemia is when you have too many red blood cells. Now, that sounds like it wouldn't be an issue, right? Too many red blood cells, too much oxygen. Oxygen's good, more ATP. Yeah, that's the good side of it. But the downside of that is the more cells in the blood, the thicker, the more viscous the blood becomes. And it makes it very, very difficult for the cardiovascular system, especially the heart, to maintain normal blood flow. The heart has to work harder. The blood pressure increases. And of course, this can cause any number of problems. The most common type, or the most common reason for the development of polycythemia naturally, is when you have a cancer of the bone marrow, and that bone marrow is being stimulated to make lots and lots and lots of erythrocytes. So we have a type of polycythemia called secondary polycythemia, and that is a normal response to that increase in altitude. So remember a while ago I mentioned that when you go up higher in altitude, there's less oxygen in the atmosphere that you're breathing. So when you have those conditions with less oxygen, then tissues are getting less oxygen. So what happens? The kidneys detect that and they secrete EPO, erythropoietin. That's going to cause more and more red blood cells to be produced so that more oxygen can be carried throughout the body to body tissues. This can, however, create that thick, viscous blood to where you may need to dilute the blood by consuming excess water, or sometimes in severe cases even having a hypotonic IV solution. But just like you would expect, too much red blood cells, too much oxygen, excess ATP. Somebody's going to figure out there might be a short-term benefit to that, right? Yep, somebody has. So increasing the oxygen carrying capacity of the blood can enhance athletic performance. So that can be utilized, especially during endurance events. Because those erythrocytes carry oxygen, athletes have tried several different ways to increase their red blood cell count, and that's what we commonly call blood doping or artificially induced polycythemia. So athletes have enhanced their red blood cell production by injecting artificial erythropoietin, such as Procrit or Epigen. Those are drugs that are used to treat anemia, so it stimulates the production of the red blood cells because it mimics erythropoietin. This is dangerous, though, because they raise that viscosity, and as we mentioned a while ago, can lead to that increased cardiovascular problems. Another way of stimulating artificial polycythemia is to remove some of your blood. So because the International Olympics Committee has determined that, yeah, we're not going to let you use these artificial EPO substitutes, right? So they've said, not a good thing. People have figured out there's another way to do it. So there are athletes that want to go about this in a little bit of a sneaky way, and perhaps they draw out some of their blood. What happens? Your kidneys detect low oxygen. Your kidneys start secreting erythropoietin. You start making lots and lots and lots of red blood cells. Then, a day or so before the event, the competition, hang that bag right back in and run that blood back in that you took out. So they draw the blood out, say, take out a pint or so. Keep it in the fridge right before the event, run it back in, transfuse it back into your bloodstream. You're creating polycythemia, right? So what's the advantage of that? Being able to carry more oxygen. What's the advantage of that? Increased endurance because of the excess ATP. As I mentioned a while ago, the Olympic Committee has decided that this is not a good thing and they have banned it, but it's really difficult to enforce because the drug is so close to being identical to what we normally produce that you can't really tell the difference in it. So you can't just check the blood to see if perhaps someone is using these substances. There is something called natural blood doping, and that seems to be the key to success to a lot of the marathon runners that come from areas like Kenya, where there is a very high altitude. So altitude training improves your fitness and endurance because your body naturally produces more and more red blood cells. By doing so, that leads to, just like we said, more ATP, more endurance. A number of training camps have even been established in Kenya, and they're attracting a lot of endurance athletes from all over the world and making lots and lots and lots of money, because you go and you train in Kenya, you develop this secondary polycythemia, and then you come back to run the Boston Marathon, and bam, lots of endurance.